This begins lesson number 4 in the cell unit for students in biology at Limestone Community High School in Mrs. Stout's class. In this lesson, we will be talking about the parts of a eukaryotic cell and also what they do. We will also describe a little bit their structure. So please take the opportunity to relate these written notes to the diagrams that you should have gotten down in lesson number 3. First of all, we'll be talking about the boundaries or the outer part of the eukaryotic cell structure. And again, let me change that color. With regards to the eukaryotic cell structure, remember, eukaryotic, we are going to be talking about organisms that have true membrane-bound organelles and nuclei. So pretty much everything except for bacteria. So plant cells, animal cells, protist cells, and fungus cells all fall into the eukaryotic category. The first boundary that we'll be discussing is the plasma membrane. The plasma membrane is the outer boundary of all eukaryotic cells, and it serves as a boundary between the inside of the cell and the outside of the cell. Its job is to allow some materials to pass into the cell or out of the cell while keeping other things out. So you can kind of think of it like a gate around your house. The plasma membrane can be described as a fluid mosaic model. And we will talk extensively about the parts of the plasma membrane in our first unit of term 2. But what we're basically looking at is fluid, meaning it is constantly moving, and it is also made of fluid particles. Mosaic, it's made of a bunch of different things pieced together. You can think of a mosaic like in art. It's kind of like a collage. And then this image is showing you a model or a representation of. As I mentioned, the plasma membrane's job is to control the movement of materials that enter and exit the cell. So not only is it like a gate around the house, but it's also, like a fence around the house, but it's also kind of like a bouncer, like the person who decides whether you get to get in or not. The big focus, the big job, the main deal for the plasma membrane is that it helps control homeostasis. So it is what is responsible for the balance of all parts of your cell. And remember, homeostasis from our first unit of information was the regulation of the internal balance in order to regulate conditions suitable for life. Some cells, not all, will have a cell wall. Animal cells do not have a cell wall. Fungus cells do, plant cells do, many types of protist cells do, but not all of them. And the protist group is kind of a catch-all garbage group. The cell wall in eukaryotic cell structures are going to be relatively inflexible, so they're going to be pretty rigid and they're going to provide support. This does show you an electron microscope image of the cell wall. It is on the outside of the plasma membrane. And here's a couple things that you'll need to know. Bacteria, protists, and plants will have cell walls composed of a substance called cellulose. Our bodies cannot break down cellulose, which is why when you eat things like corn or lettuce and salad and carrots, when you eat it and it comes out the other direction, you can still see parts of it in your exits. And this is also what's good, and this is what's in fiber and what helps keep your digestive system clean. Now, fungi have cell walls composed of a substance called chitin. It is not chitin. This one is one of the words I do want you to be sure that you're saying correctly, chitin. So you can kind of think it like a kite in. Chitin. And now moving to the organelles or the mini organs inside the cell. First and foremost is going to be the nucleus. The nucleus's job is to manage all the functions in eukaryotic cells. And as I talked about in the two different diagrams, it is going to have a couple of different parts that I want you to know. I don't just want you to know the nucleus. So it is going to have an outer nuclear envelope that is similar to the plasma membrane. However, it's going to be 4 layers thick, whereas the plasma membrane is just 2 layers thick. And then within those, within that nuclear envelope, you're going to have nuclear pores or little holes, and that's what's going to regulate movements of materials into and out of the nucleus from the rest of the cell. So stuff will go out of the nucleus, things like mRNA. And then inside the nucleus is going to be chromatin or chromatin, and these are going to be your long tangled strands of DNA. This is the form that DNA takes when the cell is not dividing. DNA stands for deoxyribonucleic acid. And a nucleic acid is an organic compound that stores information in the cell in the form of a code. And you have two different types of codes. You have DNA and you have RNA. The job of DNA is, it's kind of like your blueprint. It's your master blueprint for making proteins, which do include enzymes and your proteins for building cell parts and making cells work correctly. So this is showing you a cell. We have the outer border over here, and then this whole thing is going to be the nucleus, and this darker staining piece in the center is going to be the nucleolus. The nucleolus is what produces ribosomes, and ribosomes are involved in protein synthesis. Whenever you see this word synthesis, it just means to make. So it's involved in making proteins. Our next organelles are going to be involved in assembly, transport, and storage, and what we are assembling, transporting, and storing are usually going to be fats and proteins. First we have the cytoplasm. Cyto, from Latin, anytime you see cyto, it's going to mean cell. And then plasm is going to refer to a fluid or a gel. Cytoplasm is the clear fluid that lies outside the nucleus. It does surround all the organelles. It's kind of like a soup of material. It's like a holding medium for the rest of the organelles in the cell. We have endoplasmic reticulum, often abbreviated ER. The endoplasmic reticulum is kind of like an assembly line. It's a super folded membrane that provides a really big surface area for chemical reactions to occur without taking up a whole lot of room. This would be like if you, like when you write a letter. Let's say you wrote a letter on a piece of paper and then you needed to fit it into the envelope, so you folded it up. You didn't lose any of that letter, you didn't lose any of that room, but it's just not taking up as much space as if you sent it flat. The endoplasmic reticulum actually connects the outer plasma membrane of the cell to the nuclear membrane, and its job is to put proteins together by linking up amino acids that are dissolved in cytoplasm. So this is showing you the darker blue part here is going to be your outside of your nucleus, heading all the way to the outer cell membrane. So you can see how this is all folded in on itself. Smooth endoplasmic reticulum does not have ribosomes on it. Rough endoplasmic reticulum does. And really, they were super clever to name it this way because rough ER looks like it would feel rough, and smooth ER looks like it would feel smooth. And of course, we would have used a transmission electron microscope to be able to see the difference in the types of ER. And this would be our rough ER. The Golgi apparatus, Golgi complex, Golgi body, the Golgi, remember all of those are referring to the same thing. The Golgi's job is to collect those newly made proteins and fats and package them up and ship them either to another part of the cell or fully out of the cell. It is described as a series of closely stacked, flattened membrane sacs. So each of those membranes can be brought out when they're needed. As you can see, the job of the cell altogether is to make proteins. The endoplasmic reticulum puts the proteins together. It gets the directions on how to put those proteins together from the DNA in the nucleus. Once those proteins are put together on the ribosomes on the ER, then it's sent to the Golgi. The Golgi packages those proteins and then ships them out of the cell or to another part of the cell. So there's that. Remember, if I'm going too fast, push pause at any time. Next we have vacuoles. A vacuole is going to be like a Tupperware container or like a suitcase. It is for storage. It is described as a sack of fluid surrounded by a membrane, and it provides your storage of food, enzymes, waste, water, pretty much anything that you're going to need to save for later or save until you can ship it out of the cell or use it. In plant cells, there is one large central vacuole, and when you look at a plant cell under a microscope, it's usually the largest organelle that you can see. You'll know that you're looking at the vacuole as opposed to the nucleus because the vacuole is not going to stain. It's going to be clear, whereas the nucleus is going to have that darker staining nucleolus in it. Our next organelle is going to be lysosomes. Again, lyse to lyse, whoopsie, means to split or destroy. And these are your garbage men of the cell. They travel around the cell and they break down or digest excess or worn out cell parts, food particles, invading bacteria and viruses. So this means that they are helpful in your immune system. And these images are just showing a lysosome breaking down an old mitochondria. So they don't just go after bad stuff, they go after stuff that isn't working anymore. And this is showing them going after an invading bacterium. And that's where we are going to pause for recorded lesson number 4.